So there are two basic types of cells. We have prokaryotic cells as well as eukaryotic cells. But they are both cells, and while they have many differences, there are some basic features that makes a cell a cell. So in order to be a cell, you have to have a membrane, and a membrane is something that divides the inside from the outside, and it's going to allow the cell to regulate what comes in and what goes out of its cell. Without a membrane, a cell will die. Inside of there, we have a semi-fluid substance called the cytosol. The cytosol is the name for just the fluid. A lot of times these are going to have organelles and stuff floating in that fluid, and so you might see this called cytoplasm. So the cytoplasm would include all of the organelles inside of that fluid, and then the cytosol is just the jelly-like fluid. As far as I'm concerned, you can use them interchangeably. It's not that big a difference. A cell has to have some sort of genetic material, right? In order to be able to adapt and carry on life and reproduce, you have to have some genetic material. So in the case of animals, bacteria, this is going to be DNA. And then all cells have ribosomes. So prokaryotic and eukaryotic cells all have ribosomes, and those make proteins. Remember, it goes DNA to RNA to protein. So this protein factory is how the cell is going to carry out its genetic information. So it makes sense that it needs to have the instructions, and then it also needs to have machinery to make the products from those instructions. And we've looked at several of these kingdoms. There are two domains, bacteria and archaea, which are going to be prokaryotic. Pro here means before, and karyon means nucleus. So the word prokaryotic literally means before nucleus. And then the other kingdoms that we've seen belong to the domain eukarya are the protists, and we'll put them in quotes because they're not a true kingdom, the fungi, animals, and plants. And so those all belong to this eukarya domain because they all have eukaryotic cells. And that word EU means true, and again, karyon means nucleus. So a eukaryotic cell has a true nucleus. So here is the basic structure of a prokaryotic cell. Again, this means before nucleus. So there is no nucleus here. You can see that it does have some chromosome here. There's one chromosome, and it is kind of all bunched together in a region called the nucleoid. So here's that nucleoid region. We've got that one chromosome all wrapped up, but there's no membrane surrounding it to make it a nucleus. It's unbound. There are no membrane-bound organelles. So we're going to see a whole slew of other organelles in eukaryotic cells, like the mitochondria and endoplasmic reticulum and the Golgi body, all of these other things that involve internal cell membranes. And the prokaryotic cells have no internal membranes. So if I asked you if a cell was prokaryotic or eukaryotic, and then I described how it did not have any internal membranes, then you would be able to tell me that that was a prokaryotic cell. It does, of course, have cytoplasm. Cytoplasm is the liquid inside here. That is the cytosol and all of the non-membrane bound organelles. So, for example, those are the ribosomes. So you can see the teeny tiny dots here. Those are the ribosomes. And then the background material there, that's the cytosol. And so together, that all makes up the cytoplasm. Generally, these are going to be very small. You can see that this is on the scale of half of a micrometer. So that's 0.5 divided by a million to get to that teeny tiny number. Prokaryotic cells do often have extensions of their cell membrane or cell wall. So you can see that there's some fimbriae, they use those for reproducing. Flagella, those are going to be used for movement. And we're going to see flagella and cilia as well on some of the eukaryotic cells. So here is our generic eukaryotic cell. Over here we have a plant cell, and over here we have a animal cell. You can see that the DNA is bound inside of a nucleus, and it has a membrane. So right here in the plant cell, here's my big nucleus, animal cell, here's my big nucleus. It's always purple for some reason. And on the outside of that nucleus, there is a membrane called the nuclear envelope that is surrounding it. We see a whole bunch of other membrane-bound organelles, mitochondria, we've got chloroplasts, we've got the endoplasmic reticulum, peroxisomes, again, Golgi apparatus, lysosomes. All of those are going to contain membranes. And so none of those things will be found in a prokaryotic cell. So if I had a question that asked you to identify a cell, and I said, inside of it you saw some mitochondria, you saw some chloroplasts, you would know right away that that's not prokaryotic, because prokaryotic cells don't have any membrane-bound organelles. So these are going to be generally much, much larger, typically about 100 times larger than the prokaryotic cell. So a plasma membrane, this is one of those structures that is found in both prokaryotic and eukaryotic cells. And the plasma membrane is a selective barrier. And we will go into a lot of detail about the plasma membrane in Chapter 7. But for now, you need to know that it is semi-permeable. So it is selective. It gets to choose what comes into the cell and what leaves the cell. The cell does not just want anything coming into it. It also doesn't want to lose oxygen and nutrients and stuff like that. So the plasma membrane is there to make sure that oxygen gets in, nutrients get in, and waste gets out, and nothing else happens. Notice that the plasma membrane has to service the volume of the cell. So if you think about a cell like a circle, or in 3D it would be more like a sphere. Plant cells maybe are more like a cube. The surface area on the outside is that plasma membrane, and then the space inside is the volume. And the plasma membrane has to be able to get enough stuff in and enough waste out to be able to get oxygen and nutrients to all of the volume of the cell. And this is actually something that restricts the size of cells. So these metabolic requirements, the requirements to get oxygen and nutrients in, and to make sure that you're getting rid of waste efficiently, a cell's not going to live if it starts building up toxic levels of carbon dioxide inside of its cell volume. So those metabolic requirements set a maximum size for cells. And you can see what happens when a cell increases in size is that the volume grows faster than the surface area. So we're going to do a little math. I promise you're not going to have to do any calculating on the exam, but I'm going to use some numbers to help illustrate this. Let's draw you a little picture. Let's pretend we're looking at some kind of animal cell. Maybe we've got, it's a sphere, so here's my three-dimensional sphere shape. The plasma membrane is this outer covering, and that is the surface area. And just for knowledge, the surface area equation for a sphere is 4 pi r squared. So it's on the order of the radius to the second power. The volume of the sphere is going to be this entire fluid-filled chamber inside of here. That's the cytoplasm and all of the organelles inside of it. So the volume of the sphere is, the equation is 4/3 pi r cubed. Here you can see that r squared is going to increase quickly, but r cubed is going to increase much faster, right? If we start with r of, we can just ignore the other parts. If we start with an r of 1, well, a 1 squared is 1 and 1 cubed is also 1. If we double the radius to 2, well, 2 squared is 4, but 2 cubed is 8. So you can see how that's already increasing faster. If we increase that radius to 3, 3 squared is 9, but 3 cubed is 27. And so the ratio of r cubed is going to increase faster. So that's what this means, that the volume grows proportionately more than its surface area. So let's look at their example over here. They've got a cube that is a 1 by 1 by 1 cube. So the surface area for that is 6 times 1 times 1. And we're going to end up with a total surface area of 6, which is what they've got here. The volume is going to end up being 1 times 1 times 1, because it's just length, width times height. and that is going to be a volume of 1. So to take the surface area to volume ratio, you would do the surface area of 6 divided by the volume of 1, and you get this large number. It has a ratio of 6. So there are 6 times as much surface area as there is volume. That is going to be efficient. The cell is going to be able to use its large amount of surface area to get enough nutrients into the cell to serve that very relatively small volume. Now let's look at their second example here of a much larger cell. So this is something that doesn't really happen. We don't ever have like ginormous cells like this one here. So this one is going to have a different situation. So this is a 5 by 5 by 5 cell. So the surface area is going to be 6 times 5 times 5. So that's 25 times 6 is going to end up with 150. So that's increased a lot. But the relative change from 6 to 150 is an increase of 25 times. So the surface area when we increased from 1 to 5 increased from 6 to 150. The volume is going to even increase more. We've got 5 times 5 times 5, or 5 cubed, and so that's going to give us a value of 125. That increase is an increase of 125 times. So you can see how the surface area increased 25 times, but the volume increased much more than that, 125 times. So what happens when we calculate the surface area to volume ratio is we put 150 over 125, and we end up with that value of 1.2. And so this is much, much smaller. This cell is going to have a really hard time getting enough nutrients in through its surface area to serve this really, really large volume because the volume increased massively. So what our body typically does is it decreases the size of the cell and it increases the number. And so we end up with cells that fill up this area, but they all have a really high surface area to volume ratio.